Kumari, a postgraduate student in aquatic animal health management, and your host for this session, along with my co host Sharanya. On behalf of Women Fisheries Graduate Network, it is my great honor and pleasure to welcome you to session 3 of WFGN Global Summit 2026 titled When Water Gets Sick. Before we officially begin, I would like to invite you all to reflect a simple question. So, imagine when a person falls sick, a doctor asks, "What are you feeling?" The patient explains the symptoms, makes it easier to understand what is wrong. When a land animal falls ill, a veterinarian may not receive an answer in words, but through examination, observation, and touch, they can gather valuable clues and they can diagnose them. But what happens when a fish gets sick? It cannot tell us where it hurts, it cannot explain its symptoms. And unlike terrestrial animals, we cannot simply examine them in their natural environment. Instead, we must read the silent signals hidden in its behavior, its surrounding, and above all, the quality of the water in which it inhabits. So, this is precisely why today's session has such a powerful title, which is not when fish get sick, it is when water gets sick. Because in the aquatic world, the health of animals begins with the health of the water. To help us explore this important topic, we are privileged to be joined by 5 distinguished speakers from across the world, representing academia, research, and industry. Each of them brings a unique perspective and valuable expertise to this discussion. I encourage all of you to please participate, ask questions, and make most of this opportunity to learn from our esteemed experts. With that, let's begin with the session. Our first speaker is Professor T.S. Dr. Muhammad Effendy, ABD Wahid. He is joining us from Malaysia. He is currently Dean of Faculty of Fisheries and Food Science at University Malaysia Terengganu and Chairman of Asian Fin. He completed his degree in veterinary medicine at University Putra, Malaysia, followed by a PhD in veterinary immunopathology and was the founding director of Institute of Marine Biotechnology. His expertise spans immunopathology, vaccinology, and immunohistochemistry, making him a fitting voice to open this session on a big picture, policy, and science level. So, sir, I welcome you further to speak on the topic.
Thank you, Speaker. I will have to share my screen. All right. This is from PowerPoint. I try to make a presentation. If it's okay, then I'll proceed. Is okay now?
Yes, good.
It's in presentation mode?
Yes, sir.
Good morning, all respected lecturers, researchers, postgraduate students, and colleges. Actually, it's 7:30 p.m. in Malaysia. How are you all today? It is a pleasure to share this work with you today. My presentation is entitled PET Microplastic Toxicity in Tilapia, and I will frame it not only as an environment pollution issue, but also as a fish health and aquaculture diagnostic issue. When we speak about microplastics, we often imagine particles floating somewhere in the environment. But for farm fish, especially tilapia, The question is more direct and more clinical. Once these particles enter the body, what do they do to their physiology? Do the fish simply pass them out, or do they leave behind measurable biological stress? In this 21-day investigation, we focus on sublethal effects. That word is very important. The fish may not die, but they may still be biologically compromised. So throughout this talk, I invite you to think like a fish health diagnostician. We are not asking only, did the fish survive? We are asking what happened inside the fish while it survived. Let us begin with the suspect, polyethylene terephthalate or PET. PET is familiar to all of us because it is widely used in bottles, packaging, food containers, and many forms of single-use plastic. Its usefulness in human society comes from its stability. Unfortunately, that same stability creates a long-term environmental problem. PET resists rapid natural degradation. Under sunlight, weathering, and physical abrasion, larger plastic items gradually fragment into smaller particles. Once the particles reach the microplastic range less than 5 millimeters, it is smaller than your hair size actually, your hair diameters. They become much more mobile and biologically available. This is where the problem shifts from environmental presence to biological exposure. But a fragmented PET particle that can be transported by water, mixed with sediment, or enter fish feed is a very different concern. It becomes accessible to aquatic organisms, and eventually it may enter the food chain. So the central clinical question is this: when PET microplastics are ingested by fish, do they behave as inert particles, or do they become physiological stresses? This is slide 3. Can you see slide 3?
Yes, sir.
For this study, we used Nile tilapia, Oreochromis niloticus. Tilapia is an excellent bioindicator for this type of investigation for 3 reasons. First, it has behavioral vulnerability. Tilapia are omnivorous and often interact with the bottom environment. This makes them more likely to encounter sediment-associated particles and contaminated feed materials. Second, tilapia are physiologically relevant. They are robust enough for controlled laboratory studies, yet sensitive enough to show measurable stress responses when internal homeostasis is disturbed. Many of responses we observe in tilapia, especially metabolic and hematological responses, are meaningful for broader teleos fish health. Third, tilapia carry economic and public health significance. They are widely cultured and widely consumed by humans. Therefore, their internal health is not only an academic concern, it is linked to aquaculture productivity, animal welfare, food quality, and consumer confidence. In other words, tilapia is not merely a test organism here. It represents a bridge between environmental contamination and practical aquaculture health management. Now let me walk you through the clinical setup. The exposure lasted 21 days. Commercial pellets were crushed and mixed with high-speed blended PET microplastics. The particles were kept below 2 millimeters to increase the likelihood of ingestion and to avoid immediate rejection or excretion. We used 4 treatment groups: a control group with 0% PET, a low dose group with 5%, and a medium dose group with 10%, and a high dose group with 15%. This allows us to examine whether the biological effects were dose-related. The husbandry conditions were tightly controlled. Fish were housed individually in aerated tanks and feeding was performed twice daily. Uneaten pellets were tracked carefully. This point matters because when we work with fish toxicology, uncontrolled feed intake can easily confound interpretation. If a fish eats less, changes in glucose, growth or gut weight may reflect appetite suppression rather than toxicological effect. So the protocol was designed to separate the effect of pet exposure from the background noise of husbandry variation. This slide shows the diagnostic timeline. Before treatment, blood glucose was collected through caudal puncture to establish an individual physiological baseline. This is important because fish, like humans, vary in baseline metabolic state. A single post-treatment reading without baseline context would be much weaker. The evaluation points were day 7, day 14, and day 21. At each 7-day interval, 3 fish from each treatment group were humanly euthanized for diagnostic extraction. First post-treatment blood glucose was measured as an indicator of metabolic stress. Second, blood smears were prepared to examine red blood cells morphology. Third, the intestinal tract was dissected and weighed to assess possible gastrointestinal effect. The logic here is clinical triangulation. We are not relying on one biomarker alone. We are asking whether metabolism, blood cell integrity, and gastrointestinal condition tell a coherent story. If different systems point in the same direction, our confidence in the biological interpretation becomes stronger. The first diagnostic marker is glycemia. I think everybody knows the glycemia is blood glucose. In fish, elevated glucose is a well-established primary indicator of physiological stress. It tells us that the animal is mobilizing energy to cope with internal disturbance. By week 3, the glucose pattern shows treatment-related alteration. The ANOVA result indicates that PET treatment significantly affected blood glucose level with P equal to 0.004. In simple terms, the exposure groups were not metabolically equivalent to the control. The highest dose group showed a higher average glucose value compared with the medium and low groups, and the visual reading on the slide illustrate how fish can move from a lower baseline state to a higher post-treatment glycemic state. However, we must be very careful. Exposure duration by weeks did not show a statistically significant compounding effect, with P equal to 0.073. This means the strongest interpretation is not the glucose increase linearly every week. Rather, PET exposure itself disturbs metabolic regulation. Clinically, I would read this as evidence that the fish was spending energy to maintain homeostasis under microplastic-induced stress. The second diagnostic marker is the hematological disruption, especially the percentage of abnormal red blood cells. This is very important endpoint because red blood cells are directly tied to oxygen transport and systemic health. The data show that treatment concentration significantly influence RBC abnormalities with P equal to 0.025. But the time pattern is especially interesting. During week 1 and 2, the RBC response were relatively similar. Week 1 and 2, the response were relatively similar across treatment. That suggests that the fish were still within a phase of early adaptation. By week 3, however, we see a clearer divergence, particularly involving the medium and high PET exposure. The statistical comparison at week 3 indicates a significant differences with P equal to 0.022, specifically between the 5% and the 10% doses. This is biologically meaningful. It suggests that prolonged exposure to moderate plastics level may breach cellular tolerance. At first, the fish may compensate, but after repeated exposure, the blood cells begin to show structural evidence of stress. For fish health specialists, this is a valuable warning signal because hematological damage can appear before overt mortality. Here we move from the percentage data to the pathology behind the data. Healthy fish erythrocyte should appear structurally sound with a stable oval morphology. Under stress, however, cells become irregular, spiked. Here you can see spiked, fragmented, and show membrane disruption. These changes are not cosmetic. Red blood cells are functional cells. Their morphology affects their ability to move through circulation and support oxygen transport. When the membrane is compromised, the cell becomes less efficient and more fragile. The proposed mechanism is that PET microplastics are not biological innate. Their physical presence can irritate tissue. This may promote inflammation and oxidative damage. Once oxidative stress increases, circulating erythrocyte can become vulnerable to membrane injury. So, when we observe deformed erythrocytes, we are not just seeing abnormal shapes under the microscope. We are seeing a possible physiological cause, reduced blood quality, impaired oxygen delivery, and evidence that the fish is no longer maintaining cellular integrity under exposure pressure. The third diagnostic marker is gastrointestinal impact. The gut to body weight ratio range from 0.04 to 0.08 in the control and low dose groups, 0.03 to 0.06 in the medium dose group, and 0.05 to 0.07 in the high dose group. The descriptive trends suggest that body weight and gut weight fluctuated across treatment. Short-term high-dose exposure initially showed higher body weights, which may reflect compensatory feeding or short-term physiological adjustment, but this normalized by week 3. The key message is not that this endpoint alone proved severe gut damage. The slide is appropriately cautious. Because the sample size per week was small, these parameters should be treated as descriptive indicators rather than statistically definitive endpoints. Still, from a clinical perspective, variation in gut-to-body ratio should not be dismissed. It may point toward changes in gut structure or function, including irritation, altered absorption, impaired development, or inflammation. In fish health, the intestine is central to nutrient assimilation, immune interaction, and microbial balance. Therefore, even descriptive gut changes can help us understand the broader physiological burden of PET exposure. This is one of the most important slides in the presentation. The raw data show 100% survival across all groups, control 5%, 10%, and 15%. There was no acute mortality over the 21-day period. At first glance, someone might conclude that PET exposure was not harmful, but that would be very superficial interpretation. This is what I call the survival paradox. The fish lived, but they were not necessarily healthy. The same fish showed elevated glucose, erythrocyte deformation, and sign of gastrointestinal disturbance. So traditional aquaculture metrics, especially survival rate alone, may classify these animals as normal, while internal biomarkers reveal biological compromise. This is very relevant for aquaculture. A farmer may see no dead fish and assume the stock is stable. But if fish are metabolically exhausted, immunologically stressed, or hematologically compromised, the risk is only hidden. It may appear later as poor growth, weak disease resistance, lower product quality, and sudden failure under a secondary stressor. Therefore, survival is necessary information, but it is not sufficient information. This metric summarizes the dose-dependent pattern across the study. Acute mortality remained at 0% in all groups. But once we move beyond mortality, the internal picture changes. For metabolic stress, the control remained at baseline, the low dose became elevated, the medium dose became significant, and high dose reached a severe peak. This shows a progressive metabolic burden. For cellular damage, the control was normal. The low dose suggested mild adaptation. But by the medium dose, the fish appeared to reach a tolerant breach, and the high dose showed high deformity. This is consistent with the week 3 hematological findings. For the gastrointestinal tract, the control remained at baseline. The low dose showed fluctuation, the medium dose suggested irritation, and the high dose suggested altered absorption. The important teaching point for the postgraduate student here is a toxicant can produce a graded internal response even when it does not produce graded mortality. This is why chronic and sublethal toxicology requires biomarkers, not only survival counts. This slide brings the mechanism together. The proposed pathway begins with ingestion and retention. Hard and irregularly shaped PET particles in this study below 2 millimeters enter the gastrointestinal tract. Once inside, they may create mechanical aberration. These particles can scrape or irritate the intestinal epithelium surface, potentially causing epithelial disruption, detachment, or localized injury. The damaged tissue then becomes a trigger for inflammatory response. Inflammation is energetically expensive. The fish must mobilize resources to maintain internal stability. One consequence is increased blood glucose, as the liver and metabolic system provide emergency energy. If the inflammatory condition persists, oxidative stress may increase. Reactive oxygen species can attack cellular membranes, including the membranes of the red blood cells. This provides a plausible link between gastrointestinal aposure and the erythrocyte deformities observed under microscope. So the story is not simply that plastic enter the gut and stay there. The story is that local irritation can become systemic stress. The gut becomes the entry point, but the consequences may be seen in metabolism and blood. This chronology is useful because it explains why early results may appear mild. During week 1 and 2, the fish seems to remain within a homeostatic defense phase. Glucose may fluctuate, but the animal is still actively managing the stresses. RBC abnormalities remain close enough to baseline that differences are not strongly expressed. This is the adaptive phase. The fish is spending energy, adjusting metabolism, and trying to maintain physiological balance. By week 3, the situation changed. Blood glucose remains elevated, and abnormal red blood cells increase significantly at higher dose, especially 10 and 15%. This suggests that compensatory mechanisms are beginning to fail. In toxicology, this distinction is important. Absence of severe early damage does not mean absence of risk. It may simply mean the fish has not yet crossed its biological threshold. The more useful question is not only whether the fish is stressed today, but how long the fish can maintain compensation before adaptation becomes exhaustion. This is exactly why time cost studies are available in aquatic animal health. Now let us move from the experimental tank to the wider aquaculture system. For producers, invisible physiological stress can reduce stock performance even when mortality is absent. If fish divert energy from growth and immunity into survival, then feed conversion, growth uniformity, and disease resistance may all be affected. This matters especially in intensive aquaculture where fish are already exposed to multiple stresses, stocking density, handling, fluctuating water quality, and pathogen exposure pressure. Pet microplastic exposure may not act alone, but it may weaken the fish and make other stresses more damaging. For consumers and public health, the concern is broader. Tilapia is globally consumed fish. Evidence that pet particles can be biologically taken up and associated with internal physiological damage raises questions about edible tissues, chemical additive, and long-term food safety. We should be very careful not to overstate beyond the data. This study does not claim a complete human risk assessment, but it does support a serious warning. Microplastic contamination is not merely an environmental aesthetic problem. It is connected to animal physiology, aquaculture quality, and food system confidence. Pet microplastic behave as biologically active stresses in tilapia. The clinical verdict is straightforward. They are associated with sublethal metabolic exhaustion, shown through altered glycemia, and hematological damage, shown through erythrocyte deformity. The second verdict is methodological. Survival rate alone is dangerously inadequate for assessing microplastic toxicity. It tells us whether the fish remain alive, but not whether the fish remain physiologically sound. In fish health monitoring, we need to move forward from stronger biomarker-based approach. The third point is the future outlook. The future eco-toxicological work should prioritize longer exposure, periods, larger sample size, and more systemic biomarkers. This would include liver enzymes, antioxidant enzymes, lipid profiles, immune markers, histopathology, and tissue level microplastic burdens. For postgraduate students, this is also a research opportunity. The next generation of aquatic toxicology must integrate environmental science, fish physiology, veterinary diagnostic, and food safety. Only then can we fully understand how microplastic move from the water column into biological system and eventually into global food web. Thank you very much for your attention. I hope this presentation has shown that microplastic toxicity should be evaluated not only by mortality, but by the internal health of the fish. For me, the most important message is simple. A fish that survives is not always a fish that is healthy. When we study aquaculture species, especially species that support food security, we must look deeper into metabolism, blood, gut health, and long-term resilience. I would be very happy to receive your questions, comments, and suggestions after this, especially from the postgraduate students and lecturers. Your perspectives are valuable, and I hope this discussion can encourage further collaborative thinking on microplastics, fish health, and safer aquaculture systems. Thank you for listening.
Thank you, sir, for such an insightful presentation and for sharing this very interesting toxicology study with us. So, until today, like me also, many of us knew tilapia simply as a chicken of freshwater, a hardy fish, a proliferative breeder, and an important food fish. But I think your research has shown us that tilapia is much more than that. And while microplastic toxicity itself is an important area of research, what made your study especially interesting was how you highlighted the role of biomarkers in detecting these hidden impacts. You have shown us how biomarkers can serve as powerful tool for accessing the fish health, and also monitoring the environmental stress. Thank you for broadening our perspective, sir, and sharing your valuable expertise. It was truly a fascinating and thought provoking session. So now I will hand over the session to my co-host Sharanya, who will take us forward with the other speakers. So the question and answer session will be held at last when all the speakers will be done with their presentations.
Good evening, everyone. My name is Saranya, and it's my absolute pleasure to be your co-host for this session. I'm currently pursuing my PhD in Aquatic Animal Health Management at Kerala University of Fisheries and Ocean Studies, Kochi. And also, I'm working as a research associate at ICR Central Institute of Brackish Aquaculture, Chennai. With my brief introduction, I now it's my pleasure to introduce our next speaker, Mr. Abdul Noor Luttamaguzi from Uganda. Mr. Abdul Noor is a senior fisheries officer at Luaro District Local Government in Uganda and a PhD scholar at Makara University. He is currently serves as a global student director of World Aquaculture Society, where he actively promotes knowledge sharing, student engagement, and global collaboration in aquaculture. His research and work focus on fish health and welfare, aquaculture biosecurity, climate resilient aquaculture, and disease surveillance, with the notable contribution to the monitoring and control of tilapia lake virus in Nile tilapia. With this introduction, I extend a warm welcome to Mr. Abdul Noor to join our session. Sir, the virtual floor is yours.
Yes, thank you very much. Am I audible enough? Yes,
Yes,
moderator,
sir,
am I
you are
audible enough?
audible.
Yes, thank you for that beautiful introduction. The name of Luttamaguzi Abdul Noor, and I'm into fish health. That's my field, and then also I'm promoting the use of medicinal plants as a safe alternative to the synthetic drugs in order to control the antimicrobial resistance, in order to control the bioaccumulation in the farmed fish, which this is food that can cause public health, and also the change of the microbial organisms in the water environment in case we are to use these synthetic drugs. So among what all that have been talked about me, today let me just present part of my research. Part of my research which talks about using of medicinal plants as a safe alternative to the synthetic drugs. Currently, the Asia leads in aquaculture, and basically when you look at the utilization of medicinal plants in China, in Indonesia, in India, the Southeast countries, and also the East Asia countries, you realize that there's a great adoption of use of medicinal plants. So therefore, let me give a presentation in Ugandan context, and also using the Gyrodactylus. This is a monogenia which is prevalent in the hatcheries, specifically in the nightclub hatcheries. So, to begin with, when you look at the aquaculture production in Uganda has really grown potentials after a decade, and you realize that it has grown to, when you look at the recent SOFIA results, you realize that we're at 103 metric tons with a drop in 2002 behind Ghana. And realize this intensification has also been characterized by the establishment of new and large farms, and particularly tilapia, which is contributing about 49%, has contributed a lot to the aquatic production in Uganda. And realize this increase in aquaculture production is due to the increased demand of the fish protein and also the declining stocks from the capture fisheries. All this together has resulted in demand in the fish seed. This is part of the aquaculture value chain And when this value chain is affected, the whole production system is affected. You realize that the current seed production does not meet adequately the current needs from the farmers, and realize that these hatcheries are faced by a number of problems like poor pulp management practices, the high broodstock management. We have inbreeding. We have the high cost of feeding, the high cost of the limited access to finance. However, I point out the issue of low survival and high mortalities due to the diseases. In Uganda, I realized that different pathogens have been isolated or have been recorded in different production systems, including the hatcheries. You realize, but then in the Uganda context, the parasites have been the main disease burden. When you look at the various studies that have been made over years to the recent ones, and realize that in the hatcheries there has been a unique monogenian, which is the Gyrodactylus. This Gyrodactylus, why is it that prevalent in the hatcheries? Due to the fact that in Uganda, we always fertilize the ponds. We have the indoor and outdoor ponds, but we always fertilize the ponds as a practice in order to provide food for the fry. Yeah. And this kind of practice leads to the establishment and also the proliferation of the Gyrodactylus. And realize that these have been recorded with high mean intensities and prevalence of over 50% and 6 parasites per individual. Then realize also the recent study from me, it is 6.7% and mean intensity of 4 parasites per individual fish. So realize that this is a high prevalence and mean intensity is recorded in the specific night tilapia hatcheries. When you look at the etiology of this parasite, realize why are we having attention for this particular paradise? Why aren't we having this attention to the Dactylogyrus or the Cyclogyrus? Why the Gyrodactylus in the hatcheries? You realize that this ectoparasite is so sensitive to temperature, yeah, whereby these temperatures they increase the metabolic rate, the reproduction, and the growth of various stages. You realize that when 1 degree of increase in temperature will lessen the life cycle, and this Gyrodactylus has a direct life cycle, does not need an intermediate host. When you look at the way it's transmitted in various ways. Just contact with an infected fish, the other fish can get infected. Just when the fish is detached on the substrates in the pond or in the tanks, realize that it's effective to affect another fish. Then if the fish dies, the carcass, this parasite takes long to die, so it can be spread to another fish. When you look at the individual parasite can swim through the water column, unlike the Dactylogyrus, where they need the effective larvae, the Oncoricidium, which needs at least 4 hours to have the next host. When you look at this Gyrodactylus, it does not need in the water column, even if it's born, because this is a barrier, it will affect the other fish. Another characteristics of this Gyrodactylus, realize that it has a high transmission rate and short regeneration times. So the life cycle is too short. Within 5 days, 3 to 5 days, it's done. And then it has a characteristic infestation where it can infect the whole fish stock and reach high infestation within a short time. And this, why is it unique to the hatcheries? It majorly affects the young fish because these are a vulnerable stage. This vulnerable stage really lies on the innate immune system. Yeah. And when it is affected, this can proliferate. Then when you look at in the environment, the hatchery, how are they driven? You realize that the proliferation is driven by the high stocking density, the increased organic matter caused by the over-fertilization, and the elevation of temperatures. So combining all the etiology, combining all these dynamics of the hatcheries, you realize that this, with the current climate change, which is characterized by the rising temperatures, realize that this Gyrodactylus may further worsen and there needs urgent attention. However, what do we have available the available measures to control this Gyrodaclys, realize that different chemicals or synthetic drugs are being used. The mambazendel, the hydrogen peroxide, formalin, praziquantel, they are used worldwide to manage these Gyrodaclys. However, these ones have had some effects on the fish. When you look at mabendazole, it creates hyperproduction of the mucus on the gills, and this affects the respiration. When you look at hydrogen peroxide, it damages the gills. When you look at formalin is cancerous to the skin of human beings and also affects the respiratory system of humans. When you look at praziquantel, which is mostly used globally in order to treat these monogeneans, you realize that when you use it over time, it impairs the swimming, the respiratory distress is caused, and also these birth treatments are less effective. Why? When you apply this praziquantel, it causes hyperproduction of the mucus, and realize that these ectoparasites like the Gyrodaclys may not be washed away because the mucus has provided a protective layer and on the skin. Under the skin, this is where the parasite is, so may be less effective. So in my study, what do I propose? I'm providing a medicinal plant as an effective, biodegradable, and eco-friendly. This is a cheap alternative whereby the medicinal plants, we have the bioactive elements like the saponins, the alkaloids, we have the tannins, and these have an effect on the fish itself, on the immune system, and also on the parasites. When you look at that, they can be mixed, they can be used in isolation or in as different plants. And what do? They enhance the immune response of the fish, the adaptive immune response, and also the growth, because they have a growth parameter. They have the nutrient element in these medicinal plants. So basically, when you're applying this, it depends on the dose and the time and the method. Because basically, this can be used in immersion as a bath. You can use an injection or oral, where you can create feed additives. So in my study, I'm trying to find out if these ones can really work on this Gyrodaclys. A simple study was carried out whereby 2 hatcheries in Wakiso district in Uganda, where we have the outdoor hatchery whereby it's characterized by hapas in the ponds. As you look at this, hapas in the ponds where we do some bit of breeding, then this is transferred in holding tanks, or the partial indoor hatchery whereby fish is raised outside, but most of the part of the culture is done in the indoor. And these night tilapia fingerlings were used and they were transported to the diagnostic lab for examination. On the other side, these different medicinal plants have been were harvested. This is the neem, moringa, and Biden pilosa, the blackjack, whereby these leaves were washed, air dried for 2 weeks, and pulverized. Then extracts were made. These are the different extracts we have. Number 1, we have the neem. Number 3 is moringa, then the blackjack. And these different concentrations were made from 1 to 3 to 4 to 5, and they are stored at 4 degrees until they were used in the experiment. Then phytochemical screening was done in order to find the content, the bioactive elements that are in this phytomedicine, and how much is there, and these that are of interest that can really have an effect on the parasite. So examination was done on the fish measurement, the skin squashes, scrapings were done in order to find any ectoparasites. The gill questions were done, and the mentions were counted. Remember, in the wild, you may not find the Gyrodaclys in isolation, so you might find another monogeneans that are present. And the identification was done in the absence or presence of the eye spots. When you look at the dactyl gyrus, we have the 4 eye spots are there. Then you look at these others having no eye spots, and also the pairs of anchors. When you look at the Gyrodaclys, the anchors are prominent. We have the transverse, the 16 transverse, then the marginal hooks and the hapters, and also the reproductive organisms in the Gyrodaclys. So we had the experiment. We had the in vitro experiment whereby we had the saline solution as the control, the tank water, and 5 Gyrodactylus parasites were put in the petri dish, as you are seeing here. And then the different concentrations of neem, the neem, the moringa, and the Biden spilosa, the blackjack, were used. And the average survival time of these parasites was determined. And in order to find that this fish parasite was dead, we used a needle in order to tap on the fish in order to know if there is any movement. So primary studies realized that in the primary studies that were done in the two hatcheries, among the monogenians, Gyrodactylus was most prevalent, whereby it was 0.4 to over 1, and the mean intensity of 2 to 35 individuals in these different hatcheries. And then you realize the outdoor hatcheries, it was more prevalent than the indoor. Because when you look at the outdoor hatchery, it interacts with the outdoor hatchery interacts with the environment, whereby these fish are raised in the happers in the ponds, and you recall the Gyrodactylus are sensitive to the temperatures, unlike the indoor hatchery whereby the temperatures are controlled. Then when you look at the other monogenians like Pseudogyrus and Gyrodactylus, they had a very low prevalence and intensity of less than 3 individuals, and in some fish they were absent. We had also other co-infection of Trichodina and Ichthyophyrias were most prevalent and abundant. But our case today, we're looking at the Gyrodactylus. So when you look at the phytochemical screening, realize that in all the 3 plants, the chlorogenic acid, the necrogenic acid, and the dipyroquinic acids were present in all and in high abundance. However, in the neem, the citric acid was present only in the neem. Sorry, in the neem. So in the moringa, it's the only plant that had the furic acid and tannic acid. The absent compounds, the gallic acid and the barboscoside were present in the neem and the moringa. In the neem and the moringa, but sorry, in the neem and Biden pilosa, but absent in the moringa. So when you look at our in vitro test, you realize that the different concentrations yielded different survival times. And realize in the neem, even after the 2 hours, 1 to 4 parasites were still alive in the different concentrations. When you look at moringa, the highest concentration of 5 grams, realize that at 50 minutes, all the Gyrodactylus were dead. And the lowest concentration, at 80 minutes, all were dead. When you look at the Biden pilosa, it performed better than the neem, whereby all these concentrations of 2, 3, 4, 5, the parasites died. However, after 120 minutes, you realize that here, the Banipilosa, only one was some parasites were alive. Then in the control, whereby we use the tank water, realized that by 90 minutes, all the parasites were dead. So basically, when you look at these survival times, indicate the effectiveness of each parasite. And here, it draws our attention to what is the content in these medicinal plants. So this class realized that the Gyrodactylus was most prevalent in the outdoor, and in the outdoor because they affect young fish, and also when you look at the conditions where fish is fertilized, that's why they are high. Temperature treatments were done, though I did not present them here in the study. You can skip that. So when you look at the medicinal plants, you realize that Moringa reufela has the highest chlorogenic acids and other acids that have been seen. It alone has tannic acid and folic acid, while others are present. So realize that why Moringa performed very well. The tannins and folic acid, they interfere with the parasite neuro condition. When you look at the experiment, realize that these Gyrodactylus are always motile, they are moving. But when you apply this moringa in particular, in a shorter time, you realize that they are static, they are paralyzed because of the tannins and folic acid. When you look at the high levels of dicarfeoylquinic acids, and that one, they affect the mitochondria. You know the mitochondria in the parasite, the one that provides energy. You realize that the fish parasite will be less active, and this is due to these acids. Then, in our experiment, we realized that the extraction, we didn't find the saponins. However, it's because of the mode of extraction we used. We used methonic acid. However, when you use the ethanic acid, you can find the saponins. But however, some action was really seen. These saponins destabilize the cell membrane of the parasite, and you would see in the microscope that some granule components are being released from the parasites. So basically, that is my study, and I hope you have gotten some contributions, some insights of how the medicinal plants are providing a safe alternative to the synthetic drugs. Thank you. Back to the moderator. Yes.
Aditi. Thank you, sir. Thank you for such an insightful presentation. Your, your talk helped us to understand how climate change, particularly rising temperature, influence the parasite life cycle and disease dynamics in aquaculture. We also learned the remarkable potential of medicinal plants, not only for the treatment against the parasite, but also as a growth promoter and immunostimulant. Thank you, sir. Thank you for broadening our perspective on sustainable aquaculture. Thank you so much, sir. We will have the question and answer session later. For the next speaker, I would like to introduce Miss Lydia David from Nigeria. She is an aquaculture specialist with Child Farm Limited and has a strong academic background from Usmano Danfodia University, Sokoto. She is passionate about advancing sustainable aquaculture with particular interest in aquatic toxicology, fish health, and aquaculture production. Through her professional work, she has been actively involved in promoting best aquaculture practices and sharing practical knowledge with farmers and industry professionals. With this introduction, I, I would pleasure to invite Lydia Devi to present her presentation. Ma'am, the virtual floor is yours.
Good afternoon, everyone. So, it is a great pleasure and privilege to be part of WFG Global Interactive Summit 2026. So, I would like to express my sincere appreciation to the organizer for inviting me to participate in this important discussion. So, my name is Lydia David, and I am from Nigeria, and I hold a bachelor's degree in Fisheries and Aquaculture Technology and MSc in Fisheries and Aquaculture Management with specialization in aquatic toxicology and a postgraduate diploma in education. So my research focuses on aquatic animal health, environmental toxicology, water quality, and sustainable aquaculture. So during my master's research, I investigated the effects of botanical extracts such as Nymphaceae luteus. I worked on Leptadenia astata and urea fertilizer on night tilapia. I worked on Oreochromys niloticus. So assessing their behavioral responses, their blood parameters, I worked on their blood parameters and tissues health. And these works have strengthened my interest in understanding how environmental conditions influence fish health productivity. So beyond research, I have worked with fish farmers through aquaculture extension. So today I am delighted to contribute to our discussion on when water gets sick. So I have asked the organizer, I sent a mail to the organizer if I am to present a slides but I got a mail that it will be an interactive section. So I didn't prepare any slide. So I'm sorry for that. It is not my uh, this is so let me start from introduction from the today's topic on when water gets sick, aquatic animal health. So let me say aquatic animal health can refers to what we call physical. It can affect their physical, their physiological and biological well-being of aquatic organisms. And these aquatic organisms that we are talking about a fish, example fish. When we talk about fish, we are concentrating on night tilapia, the Orochromys ninoticus, then mollusks, and any other aquatic organisms that can be found in the water. Now, the phrase, I want to concentrate on the phrase that says when water gets sick. This phrase highlights a fundamental principle of fisheries and aquaculture in the sense that the health of aquatic animals it depends largely on the health and of their environment. That is, unlike terrestrial animals that can move away from unfavorable conditions. So aquatic animals live entirely within their aquatic environment and are constantly, exposed to water quality changes, pollutants, pathogens and environmental stressor. Now, let me say healthy water supports growth. When the water quality is healthy, it can support the growth of the fish, their reproduction, disease resistance and survival. Now let me talk about poor water quality. Poor water quality can result to what we call stress. When the water quality is very bad, it result to stress, and this stress leads to outbreak of disease to fish in the water. Then it also reduce their growth rate. You see some of the fishes, they will be having a stunted growth because of the poor water quality. Now let me talk about water quality and their parameters. Example of water quality parameters, I'll be talking the first parameters I'll be talking about dissolved oxygen, that is DO. Dissolved oxygen is oxygen available in water for respiration. Fishes in the water, they have to respirate, they have to breathe. And what are the importance of this dissolved oxygen in the water? One, it requires for breathing. This dissolved oxygen it is required for breathing. Then it also supports their metabolisms in the water. All the activities, their movement, it support their movement, their metabolism in the water. It also promotes growth. Now if there is low dissolved oxygen in the water, what is the result of low dissolved oxygen in the water? One, fish will ten d to gasp at the surface of the water. Let me say one scenario that happen. A fish farmer stock fish, and over the night, when there is a high temperature, the fish or low dissolved oxygen, high temperature leads to low dissolved oxygen. It helps to deplete oxygen in the water, and before you know, overnight all the fishes, they recorded high rates of mortality in the water. So low DO can cause stress. It causes stress in the water. So to avoid stress, there must be a good water quality. Now how can we prevent low DO in the water? First, you must fix an aerator. It is this aerator that helps to pump in oxygen, into the water. Now let me go into temperature. Temperature, which is the degree of coldness and hotness of a water body. Now temperature can affect metabolisms. When there is a high temperature in the water, it can help to affect the fish metabolisms, the activities feeding, it affects their growth. Because when the water temperature is high, the fish will not respond to feeding, so it also reduces their reproduction and disease will be susceptible. So now let me say a typical example. Tilapia generally perform well at warm temperature here in Nigeria. I don't know of other country. So in Nigeria, tilapia perform very well in a warm water temperatures. So suddenly if there is a temperature fluctuation, it can help to stress the fish and it also increases disease outbreak. So let me go into another water quality parameter, which is pH, the potential of hydrogen. Now, pH, it helps to measure the acidity and alkalinity. Now, the suitable range for mainly freshwater fish, I'm talking about freshwater fish. Now, you know, tilapia fish, the night tilapia I am talking about, they are freshwater fish. So the suitable range, the suitable pH range for Oreochromis nanoticus is from 6.5 to 8.5. So Oreochromis nanoticus can survive within the pH range of 6.5 to 8.5. And what happens when there is a low pH in the water? One, it causes gill damage. You see their gills begin to wear off. So it causes stress. And when stress occurs, stress can also lead to high rate of mortality. Now, let me go to another parameter that we call ammonia. You know, ammonia originates from fish waste. When there is high rate of mortality in the pond, first thing that we have to do before feeding in the morning, you have to remove the mortality before feeding. But when the mortality remains in the pond water, then it will lead to what we call ammonia. Then another thing is on eating food. Yes, when you overfed your fish, the uneaten food will now decompose and leads to what we call high ammonia. And what are the effects of this ammonia? It also damages the gill and also reduce their growth. So it causes growth reduction. Example, overfeeding causes feed accumulation. It will accumulate the feed in the pond and decomposes. This decomposition of feed is what will release what we call ammonia. Now, let me go to another parameter that is called nitrite. Nitrite, they enter fish blood. They enter fish blood and reduce their oxygen transport. And when this reduces the oxygen transport, one of the symptoms you are going to see is what we call rapid breathing. You see the fish in the water will begin to breathe very fast, which has a direct effect on their health. Yes. Then another one is what we call mortality. It is this stress or reduction of oxygen transport that also leads to mortality. And mortality, they are death of fish when the fish is not taken care of. So now let me go to major diseases of aquatic animals. Since we are talking about when water gets sick, what are the major diseases of aquatic animal? The first diseases that I'll be talking about is what we call bacterial disease. Bacterial disease, example is Eremoniasis, and it is caused by Eremonia species. So, and one of the symptoms is ulcer. You will see part of their tummy will block off. You will know that this fish is infected by what we call a bacterial disease. And another example of bacterial disease is what we call pulmonary disease. This pulmonary disease, some of their symptoms is you see white patches. White patches, white patches. I have a sample here. If I have samples here, so there will be white patches. There will be white patches at their mouth area here. So this is an example of bacterial infection. Now let me go to what we call pulmonary disease. This disease also affected their gills. It damaged their skins. You will see some of their skins will begin to wear off. So, and how can we prevent all this disease? One, we have to maintain a good water quality, because water quality is the foundation of animal health. So we need to maintain a good water quality. You know, biosecurity also, we need to maintain a good biosecurity. We have some of the farms here in Nigeria. They take that biosecurity very serious, in the sense that an external body cannot just enter the farm, because there is an infection from man to animal and from animal to man. So to avoid mass of the mortality, they have already done something like foot baths. You need to dip your feet before you be allowed to enter into the farm. So prevention, good water quality, biosecurity, and proper management. We need to maintain a proper management. Part of the proper management is the biosecurity that I am talking about, in order to reduce the spread of these diseases that I'm talking about. Now, let me go into viral disease. This viral disease, like tilapia lake virus, is one of the most important emerging diseases in tilapia. This is one of the virus that can be found in tilapia. It is called tilapia lake virus. And these symptoms, when they develop these symptoms, first you will see that there will be loss of appetite. They will not have appetite to eat. When you feed them, you will notice that the feed will be floating at the surface of the water because they will refuse to respond to feeding. And skin discoloration, you will see some of their skin, it will begin to change from black to pale red to yellow. So it also increases their high rate of their mortality. Now, challenges: rapid transmission. This viral diseases that I am talking about, if it is not properly handled, it can be transmitted quickly from one species to another. You know, fishes, they live entirely in the aquatic environment, unlike terrestrial animal. So the next one I'll be talking about is parasitic disease. This parasitic disease, it is caused by itch. This itch is white spot diseases, and it is caused by Ichthyophthis multifidus. So this is an example of the parasitic disease. You will see some of the worms, they will be coming out at the external body of the fish. It shows that that fish is being infected by parasites. It is being infected by parasites. Symptoms: you'll be seeing white spots, excess mucus, flashy behaviors, and so on and so forth. Now, let's talk about fungal disease. All these diseases that I am talking about, they are those diseases that are found in fish, especially the Orochromis nanoticus, that is the night tilapia. Let me go into fungal disease. This fungal disease, example of the fungal disease is saprogniasis. Symptoms: you will notice some coating-like growth on the skin. This is an example of fungal disease. There will be coating-like growth on the fish. It will look like a whitish patches on their faces. So, and it affects eggs. This fungal diseases, when it occurs or it is found in the body of fish, it usually affects their eggs and injured fish as well. And when injury occur, mortality will set in. Now, let us talk about their stress and immune suppression, how this immune can be suppressed. Now, this stress weakens immunity by one major stressors: poor water quality, handling. When you rough handle the fish will prone to what we call an infection. Overcrowding: when you overstock your fish, they will be overcrowded, and then being overcrowded, they will be struggling to feed, and before you know, they will be using their head to match another, and before you know, wound will come in, and diseases will also be prevalent in the water body. Then transport: when we are transporting our fish, in order to avoid stress, we can use an aerator bag to transport our fish. Then temperature fluctuation is one of the things that also suppress immune. Fluctuation in temperature, our weather, our rainfall, change of rainfall pattern, it can help to suppress the immune system. And what are the consequences? One, disease outbreak will occur. Poor growth: they will be having poor growth, they will be having a stunted growth because their health is being tampered with. Then mortality rate will also occur. Now, I worked on during my MSc program or studies, I worked on what we call the effects of toxicity of Leptadenia astata, Nymphaeus aelotus, and urea fertilizer on juvenile Orochromis nanoticus. And during this my MSc research, I was able to talk about the importance of toxicity testing, how we can test for our toxicity, the acute and chronic exposure of fish, their behavioral changes in the water, how this aqueous extract like the Leptadenia astata, how it affects their behavioral activities in the water, like the way they swim, the way they gasp for air, and the rest. Now, let me go into a climate change, because this climate change is one of the factors that affects animal health. Now, climate change is increasingly affecting aquatic ecosystem. These factors include what we call rising temperatures. When there is a rise in water temperature, it helps to deplete oxygen inside that water, which have a adverse effect on fish. Then harmful algal bloom. This harmful algal bloom is caused by eutrophication, which helps to increase excess nutrients in the water. Then reduce dissolved oxygen, as we know that fish need dissolved oxygen to breathe or to respirate in the water. Increase disease prevalence and changes in species distribution. So all these factors are those climate they are the climate change that affect our aquatic health. Now let me go to what we call biosecurity, because this biosecurity I am talking about is one of the most important factors to be considered when we are talking about water aquatic health management. Biosecurity, which is preventing disease introduction and spread. By we observing biosecurity, we want to stop the introduction of diseases, pests and the rest. Now measures that we can use: one, we have to use quarantine method. We need to quarantine our environment, we need to quarantine our fish equipment, we need to quarantine our farm. Then disinfection, restricted farm access. I have said it before, you cannot just allow any person to be entering your farm anyhow without observing biosecurity, because there is an infection from man to animal and from animal to man. So this biosecurity, by quarantining, or we can make something like a footpath that they will dip in their legs before entering the farm, in order to avoid the breakage of disease in the farm. Now what I am saying in essence, the key point is that prevention is cheaper than treatment. It is better we prevent than treatment, than treatment. So then in the women and aquatic health, let me talk about women, since we are organizing women fishery global summit. So women contribute significantly through aquaculture production. You see so many women, they are involved in the production of fishes, archery, grow out and the rest. And women are also involved in research, ex ten sion services. They'll be going from one farm to another to consult and to also give their advice on how they can properly manage their fish farm. You will see some women in processing. They process fish, they package and they sell, to end users, and marketing and the rest. So at this note, I would like to conclude that the water gets sick. When water gets sick, animal also gets sick. So protecting water means we are protecting our aquatic life, food security, livelihood and future of aquaculture. So thank you for lis ten ing.
Thank you, Ms. Lydia David, for your presentation for your insights today. Your discussion highlighted the vital role of water quality and the surrounding aquatic environment in maintaining fish health and enabling early disease detection. It was a thought-provoking reminder that a healthy environment is the foundation of healthy aquaculture. Thank you for your valuable insights, your patience, and for being with us today. We are truly appreciate your time and contribution. And we are also apologizing for not for informing earlier for the presentation. So sorry, ma'am. At next, we are having Dr. Abhishek Burden, an assistant professor and head of the department in aquatic animal health management at the Neotia University, West Bengal, India. Although he won't be delivering a presentation today, we are delighted to have him with us. He has kindly agreed to join the question and answer session and will answer our questions. So please keep your questions ready for the discussion. And next, we are having Dr. Sudeshna, an assistant professor and head of the Department of Aquatic Animal Health Management at College of Fisheries, Bihar, Animal Science University, India. She earned her PhD in Aquatic Animal Health Management from West Bengal University of Animal and Fisheries Science and has previously served as an assistant professor at the Neotia University. Her research interests include fish health management, bacterial disease, antimicrobial resistance, molecular diagnostics, fish pathology, and sustainable aquaculture. With the introduction, we are delighted to have her with us today to share her expertise. Ma'am, the virtual floor is yours.
Hello, Sharanya. I guess she'll be joining us in few minutes. To carry forward, we will invite our next speaker. She is Suryanta Das. Suryanta Das is a PhD candidate in aquaculture and aquatic resource management at the Asian Institute of Technology, Thailand. She holds a master's degree in aquaculture and aquatic resource management from AIT and a bachelor's degree in fisheries science from Odisha University of Agriculture and Technology, India. Her current research focuses on developing sustainable vaccination strategies to improve aquatic animal health and disease prevention in finfish aquaculture. Her broader interests include fish immunology, fish vaccination, and strategies to reduce antimicrobial use in sustainable aquaculture. She aims to advance innovation and practical solutions that strengthen aquatic animal health and support sustainable aquaculture development. Ma'am, you can carry with your presentation. Please come.
Thank you so much for the introduction. I will share my presentation. Is it visible?
Yes, ma'am. It's visible.
Okay. So, first of all, good evening, everyone. It is truly an honor to be part of this international summit and also share my journey with all of you. I am Suryasnata Das and currently a PhD candidate in AIT, Thailand. And now I am also working under a project which is part of Initiative on Innovative Veterinary Solutions for Antimicrobial Resistance under partnership between IDRC Canada and UK Department of Health and Social Care. So my research mainly focus on aquatic animal health, particularly the fish immunology and vaccination strategies for sustainable disease prevention in aquaculture. But since my PhD work is still ongoing, I cannot share my research this time. So I will just share some basic information on aquatic animal health, such as why aquatic it's very much important, how the disease develop, how we diagnose them, and why prevention, especially vaccination, is becoming increasingly important for the sustainable aquaculture. So, before we discuss the diseases, let's first understand why aquatic animal health deserves our attention. So, aquaculture is one of the fastest growing food production sector in the whole world, as we know. And today, the global fisheries and aquaculture produce over 235 million tons of food annually and supporting about more than 500 million people livelihoods. However, till now, the disease remain one of the biggest challenge which faced by this sector. Every year, we can see that aquatic disease cause billion of dollars economic losses, which affect the farmer, the industry, and the food security worldwide. So when the fish becomes sick, it is not only that the fish get suffer, including that the farmer also lose their income, the production also decline, and the international trades also get affected. So therefore, it's very much important to maintain the healthy fish, which will be essential for achieving the sustainable aquaculture. But this raises an important question. In first place, why do the fish become sick? Many people think that diseases are caused by only by the pathogen, but in reality, the disease developed through the interaction of three major factors. First is environment, then the host, and the pathogen. Some poor water quality, overcrowding, the temperature fluctuations, and also some other environmental stressors together weaken the fish immune system. And once the immunity is suppressed, the pathogen, such as the bacteria, virus, parasites, it can easily infect the fish and lead to the disease outbreak. And this is why we often say that the stress is the gateway to the disease. And by maintaining a healthy environment, we can prevent many diseases before even they occur. Now coming to what kind of disease do we commonly see in aquaculture? So fish can be affected by a wide variety of diseases. This include bacterial disease such as Aeromonas, the advercellosis, viral disease such as viral nervous necrosis, parasitic disease including argilosis or gill fluke disease, and fungal disease such as saprolegnosis. But we should understand that each disease has different cause and also it requires different management strategies. Therefore, identifying the correct disease and is extremely important before we make any kind of treatment decision. So once we suspect that the fish are sick, how do we confirm the disease? So the disease diagnosis can be understood in three levels. The level 1 is the clinical diagnosis. At this stage, the farmer observes visible signs such as loss of appetite, abnormal swimming, skin lesions, or any unusual mortality. It is very quick and inexpensive. But the problem is that it cannot identify the exact pathogen. That's when we go to level 2, which is the laboratory diagnosis. So this includes bacterial culture, histopathology, and some other serological tests which help to confirm the cause of the disease. And then comes the level 3, which is the molecular diagnosis. It includes advanced techniques such as PCR and qPCR, which provide highly accurate cause of the disease. It can detect the pathogen. So the key message here is that early and accurate diagnosis allows timely intervention and help us to reduce the unnecessary antibiotic use. Now the question comes, if the disease is diagnosed, how do we manage it? So the disease management should always focus on prevention rather than the treatment. The first line of defense is good biosecurity, proper water quality management, balanced nutrition, and use of SPF, genetically modified seed. And the second step is early diagnosis, which will help the farmer to respond quickly before the outbreak becomes severe. So the treatment should always be the final option and should be used only when it's necessary. But unfortunately, in many parts of India and all over the world, the antibiotics are still widely used. And this leads to another major challenge, that is the antibiotic microbial resistance. For many years, antibiotics have been widely used in aquaculture because they are easily available and also are relatively inexpensive. But with continuous and excessive and inappropriate use of the antibiotics has created serious problems. Large amount of antibiotics when entered the aquatic environment, it eventually contribute to the antimicrobial resistance. And this antimicrobial resistance also contribute to the environmental contamination and food safety concern. This antimicrobial resistance is now recognized as one of the major global health challenge. So instead of relying mainly on antibiotics, I think this is the time we should shift our focus toward preventing the disease before it occurs. And coming to the prevention method, I think now one of the most effective tool for prevention is vaccination. What do vaccine do? Actually, vaccine prepares the fish immune system before exposure to the pathogen. So when the vaccinated fish encounter disease causing organism, it can mount a faster and very stronger immune response. And in comparison to the antibiotics, we can see that vaccination reduces the mortality, it improves the fish health, and also significantly reduce the antibiotic dependence. Till now, I think there are various type of vaccine that has been developed, which include inactivated vaccine, live attenuated vaccine, subunit vaccine, or some nanoparticle based vaccine. And these vaccines are given to the fish through different delivery method. The different delivery methods can be injection, immersion, and oral. And this delivery method mainly depend on the fish size, number of fish that is cultured, and the farming system. As a part of my PhD at AIT, now my research is mainly focusing on developing polyvalent vaccine for Nile tilapia, where I am developing a hexavalent vaccine like I am using 6 antigens and try to develop the vaccine which can fight against the multiple pathogens simultaneously. But as I earlier told that now I cannot publish my finding. So but I can tell that through my research, my main goal is to contribute to the sustainable disease prevention strategy that will eventually reduce the antibiotic use in the aquaculture. Looking ahead, aquatic animal health is rapidly evolving. The future of aquatic animal health is becoming increasingly technology driven. There are many innovations in these days, such as nanotechnology, the encapsulated oral vaccine delivery method, biosensor, genomics, and these are helping us to detect the disease earlier and also improve the disease prevention. This technology will make aquaculture more efficient, sustainable, and environmentally responsible. And for young researchers, I think this is an exciting field with many opportunities to make some meaningful contributions. To conclude my presentation, I would like to leave you with 5 key messages. The first, we should always remember that the healthy fish begin with healthy environment. Second is that prevention is always better than treatment. Third, early diagnosis can save production and also can reduce the loss. Fourth is that nowadays the vaccination is one of the most sustainable tool for reducing the antibiotic dependence. And finally, that innovation will save the future of the aquatic animal health. And also, collaboration also will help in aquatic animal health. And by saying collaboration, I mean that as a young researcher, I believe that aquatic animal health is not only an exciting scientific field, but I think it also has a direct impact on food security, on farmer livelihood, and also environmental sustainability. And I encourage the students and young professionals to explore this field because, according to me, I think there is a tremendous potential to make a positive difference. So, with this, I'm at the end of my presentation. Thank you very much for your kind attention.
Thank you so much, ma'am, for an informative and engaging presentation on vaccination in aquaculture. Your session has highlighted the crucial role of vaccines in preventing disease, reducing antimicrobial use, and promoting sustainable aquaculture practices. Thank you for sharing your valuable expertise and giving us a deeper understanding of in this important field. We are truly appreciating your time and contribution. Thank you, ma'am. Next, I would like to call Dr. Sudeshna, an assistant professor and head of the Department of Aquatic Animal Health Management at College of Fisheries, Bihar, Animal Science University, India. She earned her PhD in Aquatic Animal Health from West Bengal University of Animal and Fishery Science and has previously served as an assistant professor at the Noor University. Her research interests include fish health management, bacterial diseases, antimicrobial resistance, molecular diagnostics, fish pathology, and sustainable aquaculture. With the introduction, I am delighted to have her with us today to share her expertise. Ma'am, the virtual floor is yours.
Thank you so much, Saranya. Am I audible?
Yes,
Hello?
ma'am, you are audible. You can proceed.
Okay. So just let me share. Okay. So thank you. First of all, thank you so much, Thoondil ecosystem and WFGN for inviting me to such a great informative session. And so today, just now I've joined, so I've seen some part of the previous presentation, which was very informative, and related to that, today I'm going to, we are going to learn regarding something which is very much related to aquatic animal health management, and especially disease diagnosis and sampling techniques. Because when we are going to prevent the disease or cure the disease, the first thing we have to know about the sample collection and to know about the diseases. So let me share the slide. Is it visible? Okay.
Yes, ma'am.
Okay, so today we'll learn about some techniques, some basic techniques for sampling, sample packaging, transportation, and monitoring and diagnosis of diseased fish. So first, I think so many presentation already has been completed. So still, just a second. Okay, here we can see that there are three circles, right? So host, pathogen, and environment. And three of them are very much important in case of disease, for disease development. So you have to take care of three of them, because if they are in a balance, then there will be no disease condition. But if there is like the host, if the host, I mean the aquatic animals, if they are in stressed condition, or the pathogen is overpowering the host, or the environmental condition is not that good, the water is polluted, the ammonia level is high, oxygen level is low, then there will be a condition, stress condition, and followed by the disease. So now what we should do when we have some news about some farm we are having disease, so what we should do? The first thing first is sampling. So sampling, there is a flow chart we can see. First we'll go for sample collection, then we have to take the sample from the farm, then transportation without any disturbance, we have to take the fish from farm to lab, then we have to go for other lab procedures. So let me clarify in the next slide. So for sampling, what other things are required? First thing, fish or any aquatic animal which you are going to monitor. So the fish, then the water sample. As I've told, that environmental condition is also very important. So for fish, the first environmental thing is water. So water sample also need to be taken, and after that we'll go to the lab. But the main thing is there are some problems in that case. In case of transportation, if the fish is already dead, we will be avoiding the sample. If the fish is live or just died, then we can preserve it into ice box, then we can take it. And before that, one more thing we have to take care, which is the history of the farm. We can ask the farmer about the details of the fish disease condition. I mean, since when the condition has been occurred, and how many fish sample we are culturing together, then any previous disease condition was there or not, any chemicals we have used or not. So these things we need to note it down. That is very much important. As we go to the doctor and they used to ask history, right? They used to take history of a patient. Just like that, you have to take the history from the farmers. Then according to the sample, you have to decide that which one you should take and which one you shouldn't. Okay, and now sampling, why we are collecting sample? If there is a sudden death condition, okay, sudden death, disease sign, clinical signs should be there. If there is an increased mortality, because sometimes 1-2 fish may die, but the rate, if it is increasing, then we are assuming there is some kind of problem. It could be environmental, or it could be normal any pathogenic microorganism can cause the disease. So another thing is regular disease monitoring and surveillance, which is some programs we are conducting regular basis, that if there is a disease case or not, because most of the time farmers do not report. If fish die, they just discard it and they do not report. So that data is very important. That according to that, we can plan our future work, or we can plan that how we can prevent those conditions. So that's why regular disease surveillance also important. That in that case also you can collect the sample. Now, what type of fish should be taken? First and foremost, it should be alive with disease sign, clinical sign. Okay. And next point is, if there is no clinical sign, then also sometimes we can see disease condition, but we should avoid any healthy looking fish. First, we will go for a fish, a live fish with disease sign. Then dead fish, or it has been dead for a long time, that is for no use. We will not take any dead fish. Okay. And especially for microbiological work. Okay. And then after that, what can be done with the sample? We can go for bacteriological analysis. We can go for virus culture. We can go for molecular biology tests. Okay, and some blood biochemistry also can be done. If fish is alive and if you can collect the blood, then you can go for blood biochemistry. Okay, so the next is preparation to collect the sample. See, whenever we are doing something, we should have some plan in our hand. Okay, some plan of work we have to prepare. You have to prepare at least 1 or 2 days before for that. Other than this, because a proper plan, half of the work will be done. Okay, with a proper plan. So what are the things you require? First of all, someone who can assist you. That will be very great, because someone should be holding the fish, someone should collect the fish, someone should dissect the fish. So generally 2 people are good to go. Next, what are the things you have to prepare? Like dissection equipment. If you have to dissect in the farm area itself, because if the fish is morbid or it's just dead, then you have to take the sample in the farm itself. So that's why you have to carry the dissection tool. Then forceps, like to collect parasites and all. So in that case, you can carry your dissection kit. Gloves, tray, paper towel, cotton, distilled water, sterile transport medium. If you can't process the sample in the lab, then you can take the sample directly from the farm area in a transport media. Then any kind of fixative like formalin, NPF, Queen's fluid. This is for histopathological analysis. For that, you can collect the sample, you can dissect the sample, particular organ can be fixed in fixative, and in fact, after like 1 week, 2 weeks also, you can process that. But if it is already decomposed while you are transporting it, then it is of no use. You should not keep it in fixative after that. Okay, so it's better if fish is dead, you dissect the sample at the farm area and it fix it for tissue or cell changes in any organs to see that. It is histopathological, for histopathological observations. Okay, next is some disinfectant, 70% alcohol. Portable microscope. If you have a portable microscope, it is very good for parasite observation. In the field itself, you can observe external parasites. So a portable microscope is very good option. Then sterile culture media, glass slide. So these things need to be ready in your lab, like sterile culture media. Okay, because media preparation takes time. So you have to do the sterilization work. So keep it ready at least two days ahead. Then anesthetics. If you are collecting blood samples or you are dissecting the fish, you have to anesthetize the fish or euthanize the fish if you are dissecting the fish. So that's why you require anesthetics, and which is very readily available in markets and which is not banned. That is clove oil, because MS222 require very low temperature. As we are residing in a tropical, I mean, India is a tropical country, so there 10-degree temperature, less than 10-degree temperature is not possible most of the areas, in most of the areas. So clove oil is a very easy option for that. So you can go for clove oil as anesthetics. Next is syringe, different size of syringe according to your fish size. You can use it for blood collection. Okay, so the next slide is about what are the guidelines of fish samples. It means how many sample you should take. So if the mortality is around 0.5% per day, then you can take 10 moribund fish, live fish or moribund fish, which is going to die. Okay, never dead fish. Okay. And next is no excessive mortality. If there is no excessive mortality or clinical sign is also not that much apparent, then you have to check 60 fish. Clear? So according to your farm condition, the sample size will vary from 10 to 60. Next, which sample will get priority? Like, of course, I'm from the very first slide, I'm telling that live sample will be should be taken. After that, if fish is just died, then you can take iced sample or any tissue in the fixative. Okay. So this is like a sample submission form or history form, which you have to take note down in your notebook, or you can carry the questionnaire. You can note down all the history, all the data regarding the fish size, fish weight, farmer's name, farmer's address, then the culture system. It is polyculture, monoculture, composite culture, or any intensive system, semi-intensive system, whatever. And also any previous disease history, any chemical has been used in the previous days. So these things, and feed, they are taking feed or not. So these things are very important. And what are the clinical signs, they are taking feed or not. Okay, so this information are very important. And also the fish species. Okay, and fish need to be documented properly, like photographs. Okay, next, how you have collected the sample according to the guideline, then how you are going to take the sample to your lab. So for that you can use, which is easy, that plastic bag, oxygen filled plastic bags, and you can keep it in a thermocol box, then you can transport the fish. And if properly packaged and insulated from heat, the container can transport fish for 24 to 48 hours without water exchange. Okay, as such you can take it for within 24. If you can reach within 24 and 48 hours, then it is fine in that packaging. Some other options are there, like insulated box with ice flakes, and gel ice is very good option. Okay, so the sample packing, transportation, and processing guidelines. So how you are going to process the sample? So live fish are preferred and should be sampled immediately. Removal from the water. So that is the very, preferable option. The next is, if it is not possible, then fish should be held on ice and it should be processed within 1 to 2 hours after collection, not more than that. Whole fish can be packaged live or freshly killed. After that, you have to keep in the ice box for shipment, and you have to reach within 24 hours. When individual tissues, if you are taking tissue samples, then it should be cold, keep in ice or any fixative, but it should not be freezed. Okay. For if you are going to do virological work, then the work need to be started within 48 hours of collection of sample, and it need to be completed within 72 hours. Okay, the cell culture need to be initiated within this time frame. Upon specimen arrival, determine the freshness of fish. So farmer generally, sometimes farmers also taking the sample and bringing to the lab. So how they do not know that how to take the sample, but you are the specialized person, you are the professional, so you have to know that if they're bringing and saying that check it, whatever, what happens. But if the fish is dead, spoiled, then there is no use of it, right? So you have to check it also, like the smell. There will be a pungent smell, a spoiled smell. Then appearance of eye, it will be white and opaque. Gills will be white. Internal organs will be liquefied. Okay, and post-mortem stiffness, I mean rigor mortis will be there. So these things you need to check. If it is there, then you have to tell them that, bring any morbid fish or live sample, then only I can diagnose the disease. Otherwise, what happens, that the environment is full of different kind of microorganisms. We are not alone living in the environment, right? There are so many microorganisms around us. Some of them are spoilage bacteria who only degrade any kind of vegetative cells or dead samples. I mean, fish, human, whatever. They are after death, they will be degraded, and that will be accelerated by some kind of microorganisms. So sometimes what happens, if we're processing a dead fish for which is dead for a long time, it will be contaminated by the environmental other microorganisms, spoilage microorganisms, which will confuse your data. You will get the environmental microorganism and you will think that it is the causative agent of the disease, which will be very wrong, okay, and misleading. To avoid these possibilities, we are always prefer the fresh sample. Okay. If shipping is delayed beyond 24 hours, some tissues may not be suitable for processing. Okay. And of course, before dissecting, I've told that euthanization is important. And in that case, clean a sterile dissection instrument like scissors, whatever you are using, wash it with alcohol. So that will be better. And also disinfect between the sampling of each fish species. Like there are 2 samples with you. So once you are handling, after that wash your hand and disinfect your. I mean, you are wearing the gloves, I know, but still, if you are not changing the gloves, then you have to wash your hand with some disinfectant. Okay. Next, let's come to the common gross and clinical sign of diseases. Like, what are the clinical sign you will get when you are going to sample some fish? First thing, the farmer will tell that they are not taking feed, that is loss of appetite. Then this skin discoloration, scale loss, skin peeling, pale gill discoloration, gill whitening, then anoxic condition. I mean, they will be gulping near the surface. Erratic swimming or slow swimming or lethargic movement. Then they will rub their body against the pond bank or tank surfaces. Then excessive mucus production, that is also a sign of some difficulties and stress. Less ulcer or any kind of hemorrhages on the body surface, eye, internal organs, or normal surface area you will see. In fact, after dissecting the fish, you can see the hemorrhages in their peritoneum cavity as well, or other organs. Okay. Liquefied internal organs, fluid accumulation in the body, then discolored internal organs, then gill rot, tail rot, fin rot. So these are some common sign which will indicate that the fish is diseased. So there is we can see how we are going to check the fish. First, we can see that first one, how we are checking the fish. First, you can see the first box, fish dissection. And after the second, you can see we are wearing the gloves. And then after that, we are checking the fin, then other anterior surfaces, then different parts, then mouth, then gill, then eyes, every external surfaces. Okay. After that, we are going to dissect and check each and every organs like liver, kidney. So, and one more thing, I just wanted to say that if there is a bacterial disease, always the target organ will be kidney. Okay, so that is important for bacterial diseases. Next is for external parasite analysis or internal parasite analysis. For that, we can make aqueous sample and we can observe it under microscope. Other than this, for water sampling, we can take a water sample for temperature analysis. What are the temperature, what is the pH, dissolved oxygen level, alkalinity, hardness, turbidity, ammonia. So these are the some common things which need to be, these parameters need to be analyzed. So now we are coming to end of the slide where if you are seeing a sample, diseased fish sample or dead fish, so there can be three kind of possibilities. Okay, one could be water quality is compromised. Another one could be infectious disease, and another one could be any toxic substances could be there in the water. So if you will merge them, like there will be a non-infectious diseases or infectious diseases, that way also we can classify it. Now just follow the flow chart. If water quality is not good, the first thing you will do the test, you will check the parameters of ammonia, dissolved oxygen, pH, everything, alkalinity. Then if it is normal, then go to check for any infectious diseases there or not. If it is abnormal, the water quality is poor, then you have to go for water exchange and you have to change the water, you have to improve the water quality. If there is an infectious disease, then you have to check the clinical sign, which is matching with your disease information or not. You have to check that. And also you have to take the sample to the lab for further bacteriological analysis, virological analysis, or parasitological analysis. And also fungal disease diagnosis. And if there is any toxic substances, then sudden death will be there, first of all, because toxicity, if somebody puts some toxic substances in the water, then there will be a sudden mass mortality, which is a significant sign, because there will not be such much clinical signs. Okay. So then you can check the water for toxicity. You can send the sample for toxicity test or dead fish for the toxicity test. So this is the overall sampling criteria and overall simplified version of how we are going to do, what we are going to do your samples. So I think something has been understood, and thank you so much for your attention. If any questions or anything anybody need to know, if any questions or anything anybody need to know?
Thank you, Dr. Sudeshna, for your insightful presentation. You have clearly highlighted the proper sampling techniques, like proper preservation, collection and storage, and transportation of the samples, for which is fundamental for the accurate disease diagnosis. We truly appreciate you for sharing your expertise with us. Thank you once again.
Thank you for your words.
Moving on to the next session, I would like to thank you all, our distinguished speakers, for their insightful and engaging presentation. We will now begin our question and answer session. I invite all the participants to post your question in the chat box or use the raise hand features if you would like to ask your questions directly.
Hello.
Yes.
Okay, as the participant posed their question, I have few questions for our speakers. The first question is with Abdul Noor Luttamaguzi. So, the first question is so based on your study, is there any specific temperature range at which the gyrodactylus becomes more severe in fish? Hello, Abdul, Noor.
Say again.
Yes, sir.
I didn't get the question. I didn't get the question very well.
Yes, sir. I may repeat the question. The question is, so based on your study, is there a specific temperature range at which the gyrodactylosis becomes more severe in fish?
Now, you are getting me very well. Now, when you look at these gyrodactylus, we have different species. We have the ones of the tropics, and one of temperate. When you look at the range, these can survive even at 40 degrees Celsius, the ones of template. Then we have ones of
So actually, you are not audible. Hello, sir. You are not audible.
that survive even at certain areas.
So, here am I audible? Oh,
Yes, sir.
sorry.
It's audible now. It's better than that.
Yeah, I'm audible. Hello?
Yes, sir. Please speak.
Yes,
You are audible.
I'm trying to.
Okay, sir. It seems that you are in some weak network. Please reconnect and join with us. I can see one of our participants, Frederick, is raising hand. And if you need to ask something, you can, please.
Yes, thank you very much, and good afternoon, everyone. I will wish to ask a question to the last speaker, that is Dr. Sudeshna, about her diseases. Actually, she had a very wonderful presentation. As we all understand that fish diseases plays a very critical role in aquaculture, and it's diseases that again leads to lots of mortality. Now, just to help most of the farmers who are upcoming, because again, you are interested probably in the developing countries. We understand that disease prevention is very, very important as being effective and economical than disease treatment in aquaculture. Now, some of the realities we are considering faced by farmers in developing countries is now the cost implications that they incur in curbing these diseases. Now, maybe on your own perspective, what are the possible or affordable biosecurity measures that maybe you could to use them as proactive measures?
Biosecurity measures, see, what the first thing we need to teach the farmer that one thing is that using, without any regulations, without any guidelines, they are using antibiotics and chemicals. So that need to be stopped, because medication in case of fish disease, it is very tricky, because most of the cases, most of the medicine do not work. If fish have started to die, they will die. And if we are using high amount of chemicals and without any, like for parasitic disease, farmers are using antibiotics without knowing the actual cause of disease. So we should, first of all, we should give them some kind of training or field level demonstrations that will be helpful for them, to let them know that what are the medications or chemicals are FDA approved and in which temperature. As I've told that some chemicals are approved, FDA approved, but still in our temperature condition, it is not permissible. Okay. Then like that, we need to give there the information, what things we should not do. And the aquaculture people will give them that how we have to make the pond, how we have to, how many stocking density should be there. So these things will be fine, but to prevent the disease, first thing we have to reduce the use of chemicals. The second thing is, if there is some information about disease, any clinical sign, if they are observing, they should inform the nearby professional who all are, who have some knowledge regarding this. And also, if fishes are dying, they used to keep the fish as such in the pond. They are not discarding the fish properly. So that is another cause of outbreaks. They are causing diseases to other fish. So that is another thing, that they should know the proper biosecurity process. So for that, some training or field level demonstration should be there. One more thing is that quarantine facilities, most of the farmers, they bring the seeds from any hatchery or some places. So they do not know about the history, any diseases are there or not in the seeds, or any fish they are bringing fingerling, fry, whatever. So they need to be kept in the quarantine facility for some days before introducing to the main pond. So there's some measures need to be taken. And for big level farms, so there must be some proper biosecurity, like any transportation, any vehicle is coming with fish, that also need to be cleaned, disinfect. And also the farmers need to clean all, whatever they are using, nets, buckets, whatever things they are using for disease sample, that need to be disinfect every time when they are introducing those instruments in other ponds. So that will prevent the disease spread. So these are some few points, I think, that these things are important, which the farmers should know, and that way they can maintain. And also that monitoring, because they are giving feed every day to the fish, so they are the first one to check that if anything is there or not. So any abnormalities mean they should inform the nearby professionals, or before introducing any kind of ABCD chemicals. Okay, so that is, I think, important to control the disease and prevent the disease.
Okay, thank you.
Thank you, ma'am. Now I can see Abdul Noor is available. Will you be now able to answer our question? Abdul Noor, could you hear us? Okay, we next move forward.
Hello, Aditi. This
Yes,
is
sir.
Dr. Abhishek.
Yes, sir.
Yeah, regarding this Gyrodactylus, no, that question that was being asked.
Yes, sir.
So this Gyrodactylus and Dactylogyrus, these both are tropical flukes. And the problem with that is there's nothing such like particular temperature ranges. Since it's tropical thing, so maybe during the warmer conditions only.
Species and yeah, then
Yes, sir. Please continue.
So your question was then, is there any particular temperature ranges?
Yes,
As such,
sir.
as such, there is no particular temperature ranking. Of course, different researchers, different scientists have given in their articles, in their findings, they've given different ranges. But basically, altogether, if you go in a gist, they prefer tropical climates, mostly tropical climates. So I think the tropical temperature, I think, is the best suitable for infection of these flukes.
Yes, sir. That aligns with Abdul Noor's research part also. He's also from Africa, and I think both countries have similar climatic conditions that may be the reason of that Gyrodactylus prevalence there also.
Yeah.
The next question I would like to ask with Sir Mohammed Effendi. The question to you is that what is the specific toxicological effect of PET microplastics compared to other microplastics, which are generally uh, seen, like polystyrene or polyethylene. Could you hear me, sir? Hello?
Can you replace it? Replace the
Yes, sir.
question?
Yes, sir. Yes, sir. So, what is the specific toxicological effect of PET microplastic compared to other microplastics, such as polystyrene or polyethylene?
I think different plastic have different toxicology effect. So I only study PET because I want to see whether this PET has some toxicology effect to the fish. So I was only focus on PET.
Okay, sir. So, we have like seen the toxicological effects of BPA also. That also includes in
Maybe
plastic.
somebody can do another
Yes, sir.
study by using another type of plastic.
Yes, sir. Yeah, sure, sir. Sir, my personal question to you after going through your presentation is that as tilapia is a proliferative breeder, we have seen, and you have done so many hematological parameters and everything tests and all. So, is there any changes in the reproductive biology also on the fish
Some
after this
change in
toxicology?
reproductive. So we study only for 3 weeks.
Okay, sir.
So we don't study until they come to the stage of reproduction or growth. So we just have the after the fingerlings.
Okay, sir. Okay, sir. I can see your message in the chat box. This is to all the postgraduate students. Sir, like to search you about behavioral fever in fish as one of the methods to control unspecific disease.
Yes, because behavioral fever is becoming increasingly important in fish health monitoring because it can provide one of the earliest observable indicators that fish is mounting an immune response. So because to mount an immune response, you need to have temperature, right? Because fish is ectothermic. They are poikilothermic. So they don't have any, they don't produce any temperature from inside their body. So different than human, we can produce temperature. We can regulate temperature from our inner body, but fish cannot. So they depends on warmer waters. When they are feel sick or unhealthy, they will swim to the warmer waters. So by increasing, if you have the tara, where the fish can go to the passage where the warmer water is there. So the fish that located at the warmer water is the fish that is sick. They are unhealthy. So that is one early biomarker of disease. And it also reflects immune system activation because elevated temperature enhance immune function. Numerous studies have shown that moderate increases in body temperature can increase leukocyte activity, improve phagocytosis, stimulate antimicrobial peptide production, accelerate antibody development, enhance cytokine signaling, and reduce pathogen replication in many infections. So it is unspecific. It is very broad. You can use behavioral fever mechanism to improve disease diagnosis and also to support non-invasive health monitoring and also to control AMR. I can see AMR in one of the speakers' slide just now. So that can avoid AMR as well.
Okay, sure, sir. That's something very new and interesting. One question is asked by Sagya, sir. This question seems to be very relevant to each of the speakers. I would like to invite every speaker to speak on this question, which is, can vaccine completely replace antibiotics in aquaculture? What are the scientific limitations? I would like to request every speaker to please give their insights on this particular question.
See, Aditi, vaccine, it depends that which kind of vaccine, first of all. And vaccine is a very disease specific. Vaccine is specific, okay. Vaccine is not like general, not a profile. I mean, general treatment. It must be a disease specific, and which is the everyday. I mean, very common diseases are bacterial diseases, which is a secondary pathogen. And for secondary pathogen, vaccine production is, not that feasible, because at a time there will be multiple bacteria. Okay. So these things are a bit challenging in that case. Other than this, for viral infection and all, that is possible, but still again it will come to the how we are applying the, if it is through feed or if it is through immersion, that is easy, but through injection, which is I mean, absolutely not possible, not feasible for farmers. So which kind of disease it is, that is important.
Ma'am, actually, one second. For humans, we have many diseases, many bacterial outbreaks. For fish, when it comes to the fish, there will be very limited disease, right? When it comes to the fish, there will be a bacterial disease, predominantly around 4, 5, 6, maybe 6 diseases for the bacteria. But why the vaccination is not developed yet?
Vaccination, the application, I mean the administration of vaccine, that is a first and foremost challenge. And you can see that vaccine for human also, vaccine is available for certain viral infection, not most of the common bacterial infection. Okay. And also for fish, secondary infection is very common, because fungal infection, then primary viral infection and parasitic infection, these things are primary. Other than this, most of the bacterial infection are secondary. And at a time, there will be so many opportunistic pathogens, so vaccine will not work sometimes. And
yes, please.
I would agree with that opinion, because I think in vaccination, we are far from establishing a good vaccination strategy for aquatic species, because vaccination is very specific, like what you say. And if the pathogen strains are differ from the vaccine strains, it cannot protect the fish from that disease. And fish are easily stressed. So when they are stressed, their immune system is compromised, and then the vaccine will not work as we expected. So there must be a lot of control to vaccinate aquatic species. So right now, I think we need more study on the best method to vaccinate the aquatic species, and also control, let's say, water quality, temperature, and also other environmental factors that can affect the effectiveness of the vaccine.
Yep. According to the Suryasnata Das, she tried multiple, innate immunity studies about vaccination. Can you please answer us, Suryasnata Das? Because your research is more involved on vaccination in IIT Thailand. Suryasnata Das, are you available?
Suryasnata Das, ma'am, can you hear us? Hello? Ma'am, can you hear us? Okay, she's not available, I guess. Abhishek, sir, would you like to add?
Yeah, I want to just add one point to all the speakers, those have said those things. I completely appreciate the response also. But another thing is that vaccines and antibiotics, of course, they are both chemical-based treatments. Of course, they are both therapeutics. The thing to be remembered is that vaccine is generally a prophylactic measure. That means in aquaculture or in fisheries as such, in the lower vertebrates, even in the higher vertebrates also, vaccines are generally used as preventive treatment. Like we take a booster dose, we take a dose and just pray to God that the vaccines will work. The same goes for aquaculture also. If you're giving vaccines, it is during the quarantine process or after they're being stocked. So these are basically a preventive measure. But the thing is, antibiotics are a therapeutic measure. Antibiotics are applied when the disease outcomes start showing. We do not like start the antibiotic course once the fish is stocked. We see the disease symptoms are there. What are the symptoms? Either it's bacterial, then only we'll go for antibiotics, or if there are water quality parameters, then we'll just avoid antibiotics and focus on the pH and all those things. So antibiotics are especially used in aquaculture as therapeutic measure, whereas vaccines are used in aquaculture for preventive measure, prophylactic measure. So the thing is, as per your question, that vaccines cannot completely replace antibiotics as such. There are in the European countries, vaccines are being formulated, they're being applied by immersion, by injection, of course. But still, when if infections start showing up, then after vaccines, even if they're given, antibiotics are our last hope. So that's a reason why vaccines and antibiotics have to be both applied together in some cases. There are several scenarios in European sea bass or in salmon aquaculture where vaccines are also given. After that, also their disease outputs. Then antibiotics are to be given. Of course, due to the AMR, this antimicrobial resistance, vaccines may or may not work. So new generations of antibiotics are going on coming. Like 8 generations are also now available. Even colistin is sometimes showing resistance, some bacterial pathogens. So the thing is that both has to correlate, of course, at the appropriate dosage. And personally, I don't think that vaccines will completely dominate or completely replace antibiotics. Both have different mechanism of actions and both has to be there for the sustainability of aquaculture. Okay.
Thank you, sir.
Yes, sir. Thank you, sir.
And Aditya, one more question you asked now regarding this microplastic toxicity in reproductive behavior.
Yes.
As such, I had some colleagues or I had some work done on the microplastic toxicity. The main thing with microplastics is that when it enters the physiological process, the main problem is that one main problem is that it leads to atresia. So it basically targets this germinal cells in the Leydig cells or those things, and they ultimately affect the fish breeding pattern or behavior. Of course, in the long term, there may be changes in there may be some chances of polyploidy also. Some kinds of genetically different organisms may pop out, like in the F1, F2 generations. But if you go on an overall aspect, microplastics, it affects the cells. And as such, the cells, if it attacks, the whole behavior changes. So that's the main problem of microplastic toxicity in aquaculture. Like if you're going into the only breeding part.
Yes, sir. Actually, the thing was he was doing the research on tilapia, and as we know that tilapia is a proliferous breeder. It is known for that actually. So that is
Yeah.
why I was asking about the thing that after doing the research and all, where there was something related to reproductive biology or not. So now I got your answer. The same may be happening with that fish as well.
Yeah, the thing with tilapia is that tilapia is such a prolific breeder that monosex came into existence just because of that only. But still, after
Yeah.
monosex, or even if it's coming also nowadays, but that also there are some research showing that the tilapia it changes its sex reversal. So there are mature tilapia also going under sex reversal. So tilapia, like we cannot control as such. This is the breeding of tilapia. Of course, microplastic, if you affect like commercial, like if you're going for commercial aquaculture, microplastic will pose a very huge threat. Not only now, in the long run also, as the plastic usage is increasing, there are a lot of problems with like if you leave the fishes out, there will be planktons also, which will be problematic in the long run.
Yes, sir, rightly said. Anything in excess is poisonous.
Hmm.
Okay, so thank you, sir. My next question is to Lydia. Lydia, ma'am, are you there?
Yes, I'm available.
Ma'am, we have one question for you. What routine water quality monitoring practices would you recommend for the farmers to maintain a healthy culture environment?
Repeat the question, please.
Okay, ma'am. What routine water quality monitoring practices would you recommend for the farmers to maintain a healthy culture environment?
Okay, thank you very much for the question. So one of the management practices I would recommend is one, first is to maintain a good water quality. Then I will also recommend daily checking of water quality parameters such as dissolved oxygen, temperature, the water temperature, and pH. And also there should be a way of you putting in biosecurity in order to avoid the introduction of diseases in the farm. So there should be proper quarantine, disinfection of aquatic equipment and the rest in order to avoid the spread of aquatic animal diseases in the farm. Is there any other question, please?
Thank you, ma'am. I have one more question. What is your ideology on AI on water quality monitoring for the regular pond management practices? Can you share your idea on how the AI or how the computer-based monitoring can be useful for monitoring the water quality parameters regularly?
Can you please repeat the question?
How AI can be used to monitor the water quality parameters regularly in the ponds? Can you give some idea of yourself?
Okay, you mean the idea of before checking the water quality in the pond? Okay, some of the ideas I can share is I've mentioned it. Maybe when you are running a farm and in the morning you notice that those fishes in the pond, they are gasping for air. So it shows that oxygen is being depleted in the pond. So there is a need for you to, first method you can apply is you can drain out the water completely and impound another fresh water, or you can install what we call an aerator in order to supply more oxygen into the water. So those are the first step to be taken before you can check for the water quality parameters in the aquatic environment. Thank you. Is there any other question? Is that answered?
Yes, ma'am. Thank you. And the last question is, h- how can researchers and students from India and Africa work together to advance the aquatic animal health and promote sustainable aquaculture? I request all the speakers to give an idea about this. I will repeat the question. How can researchers and students from India and Africa work together to advance aquatic animal health and promote sustainable aquaculture?
Okay, thank you very much. So how can Nigerians and other countries work together to promote aquaculture? First, we need to collaborate on something like research and innovation. There should be a kind of joint studies on example like what Professor Mohammed have talked about, the macroscopic impact on fish species like tilapia. He has talked about tilapia, the nine tilapia, the Orochromis nanoticus. So I would like prefer if there will be like a joint or research and joint innovation so that we can brainstorm and share ideas and how we can move forward about further research. Then another one, there should be a kind of development of sustainable feed additives. Those sustainable feed additives, example like the plant-based products of a thing. So then another thing is we should always be ready to share experience or expertise in disease management. Like there are some other disease management that could occur in India and here in Nigeria. We might not be experiencing, but we sharing ideas together, we'll be able to know that yes, there are other disease outbreaks occurring in other foreign countries. Then another thing is that we can do like a capacity building, in the sense that we can exchange programs for farmers. Yes, we can organize some programs for farmers, students and research. So by building this capacity building, it will help trainers, to join training seminars or on the aquaculture practices. So in conclusion, so dis benefit of collaboration, it will help to provide what we call food security. Yes, it will help to provide food security. Then it will help to provide what we call economic growth, environmental sustainability, global influence. From there we'll be able to connect with each other. Yes, so as in Nigeria to India partnership in aquaculture will not only improve fish farming practices, but we also strengthen what we call food security, trade and environmental resilience. Thank you very much for the question.
So, as we come to the end of this session, we have had an incredible journey through all the speakers listening you. Today, we explored everything from disease sampling and diagnosis to antibiotics, antimicrobial resistance, microplastic toxicology and biomarkers, climate change, parasitic dynamics, medicinal plants as antiparasitics, growth promoters, immunostimulants, the importance of water and aquatic environment in disease detection, and finally, the vital role of vaccination in disease prevention. Together, these talks have reminded us that aquatic animal health is not just about treating the diseases, it is about prevention, sustainability, innovation, and protecting the entire aquatic ecosystem. Thank you to all the speakers. And as we come to end of the session, I hope today's discussion have helped and in gaining knowledge. So, on behalf of Women Fisheries Graduate Network, I extend my heartfelt gratitude to our distinguished speakers for sharing their knowledge and experience, and to every participant who has been participating us since afternoon from around the globe, and for their enthusiasm, thoughtful questions, and their active engagement. I would like to leave you with one final thought, that when we care for our waters will always care for us also. So every healthy pond, river, lake, and ocean begins with informed minds and responsible actions. So continue to stay curious, keep asking questions, embrace opportunities to learn, and never stop exploring the incredible world of aquatic science. After all, the world is your oyster. So thank you and have a wonderful day. We hope to meet you tomorrow at the session 4 at 1 o'clock IST and 7:30 UTC. Thank you.